All right, let's begin. So my name is Lisa Sipenko. I head Consilium Scientific, and today we have a fantastic panel for you to talk about very, very rare diseases, N-of-1, and we'll get the definition of what it means, and not just the development of solutions for patients with very rare diseases, but actually taking these solutions to patients all over the world. So I would like to ask the panelists to introduce themselves, and let's begin with Becca, please.
Hi, I'm Becca Ahrens-Nicholas, and I'm a metabolic physician and clinical geneticist at the Children's Hospital of Philadelphia and University of Pennsylvania. So what that means is essentially I'm a rare disease pediatrician and physician. In addition, I'm a researcher and scientist at CHOP and Penn, and I run a translational research lab trying to develop new gene replacement and gene editing therapies for kids with ultra-rare metabolic disorders. I have the privilege of participating in a number of efforts of trying to advance therapies for ultra-rare diseases and for N-of-1 cases. So I'm part of something called the Bespoke Gene Therapy Consortium, which is an effort in the United States to try to build a roadmap for how to develop AAV gene therapies for rare and ultra-rare, really ultra-rare disorders, so that other ultra-rare communities can learn from our experience. And then in addition to that, I'm part of something called the Somatic Cell Genome Editing Consortium of the NIH, and this is an effort focused on advancing gene editing therapies into the clinic. And as part of that work, we're trying to develop N-of-1 gene editing therapies for some of the patients I care for with severe, devastating metabolic diseases. And this includes a recent effort to develop a personalized base editing therapy for a little baby that had CPS1 deficiency. And I'm happy to speak more about that. But in general, my goal really is to try to move the amazing technologies that are being developed at the bench into the clinic in a way that we can actually evaluate safety and efficacy and then try to expand to many, many cases of N-of-1. So thanks.
Thank you, Becca. Winston, please introduce yourself.
Hi, thanks so much. Winston Yan, I'm a physician scientist as well as a gene editing drug developer. I have really two hats that I wear. One is as in industry, where I'm the co-founder of a clinical stage CRISPR gene editing company called Arbor Biotechnologies. Over the 8 years of our existence, I've had the privilege of taking our first treatment, or sorry, investigational treatment, I should say, from bench to bedside. And then perhaps the other hat that's most relevant here is that I am also one of the founding team of the N-of-1 Collaborative. We're a global consortium of scientists, patients, drug developers, everyone who's interested in enabling this field of individualized medicines, or sometimes we call it interventional genetics. We do this by enabling. We do this by sharing resources, hopefully creating a common playbook. So even though all of these N of few treatments, they may be different, and the diseases they they try to treat are different, the overall data collected, because we're using a shared playbook, they can be more alike than different, and we're able to have learnings and on safety and efficacy get shared back with future treatments, so that there's not a lot of reinventing the wheel. So thank you.
Thank you, Winston. Dan, I would like to give you a word.
Yeah, thanks, Lisa. Afternoon, good morning, everyone. Really great to be here. Such a great topic. It's so topical now to be talking about N-of-1 or N of a handful. So I'm Dan O'Connor. I work at the Association of British Pharmaceutical Industry, Director of Regulation and Early Access. And before that, I was at the MHRA in a variety of different roles. That's the UK medicines regulator. And before that, I had a clinical academic job. And my interest here really is that I truly believe that regulation can be really innovative and really help move the field forward. And I think without a proportionate regulatory system, you really struggle to hang off the rest of the aspects around creating an end-to-end pathway. And I think, the science is so tantalizingly close to the patient now. We've just got to somehow bridge that disconnect between the patient and the science. And I think regulation has a huge part to play in that. And hopefully we'll get into that in the discussion today. Thanks a lot.
Thank you, Dan. Carol?
Hi, everyone. I'm Carol Longson. I'm now an independent advisor in health technology assessment and market access Professor of HTA at Manchester in the UK. For a very long time, I worked at NICE. I was the executive director responsible for all of the technology appraisals and other technology evaluation programs. That included the highly specialized technology program, which I established with some other expert colleagues at NICE. And that program looked to develop bespoke methodologies and processes for the evaluation of very rare, so we call them ultra rare diseases. I think we might have had an N-of-1 or maybe an N-of-1 or two going through that program. It brings up all sorts of issues. That type of evaluation brings up all sorts of issues, not just about HTA, but about how our system needs to be configured to be able to deliver something like that, and how evidence generation can happen to the in a way that allows, again, a very, very rare set of evidence to be used in a very robust way. So lots and lots to talk about.
Thank you, Karen. Last but not least, Duran, please.
Thank you very much. It's a real privilege to be here. I know many of these people by reputation, but some of them by historical connections a while back. N-of-1 Rieger, President and CEO of the Canadian Organization for Rare Disorders. I was just finishing my 9-year term as chair of Rare Diseases International. And one of the things we're saying is at this last WHA assembly, we actually passed for the first WHO resolution on rare diseases. So really looking forward to how that might actually have some impact in terms of global rare diseases. I'm also president of the Asia Pacific Alliance of Rare Disease Organizations. So I spend a lot of time working in the Asia Pacific area and trying to span what are really highly developed countries as well, Australia, Japan, Korea, but also really have a special interest in what we can do in terms of bringing in some of the middle income, lower middle income countries like Malaysia, Thailand, Indonesia, and the Philippines into the rare disease space. So really excited and about the opportunity to discuss this. And also, I think really wanting us to have some opportunity to talk about, and so what about? What about the rest of the world? What about lower middle income? How is this going to help us? And I think I've heard some of the strategies, but I do think that as well, we recognize that. I think we say oftentimes, the more and more we get advanced, the more and more we risk having a greater divide between those at the very top and those that are really just trying to come into it. So really looking forward to this conversation. Yet we know that the N-of-1 could be particularly relevant to many of our patients that are really living in some of these remote areas.
Thank you. Thank you so much for these introductions. So I would like to begin by giving a word to Winston and Rebecca to take us through the definition of what it means, the N-of-1 disease. What are the current developments? Where are we from the technical perspective?
Fantastic. So I have the opportunity to try and set the stage, and I understand that this is a very diverse audience for the Consilium Scientific, so I'll do my best to frame both the challenge as well as the opportunity, and I'll pass it off to Becca for some of the, what is actually happening. So ultra rare, rare, you've probably heard of these definitions. I'll just say some facts. I think if you look at the exact definition of rare disease, it's something that is less than 1 in 2,000 people have rare disease. So I think functionally that in the US definition, that means like 200,000 people and fewer have the disease. And then ultra rare, it's a subdivision of that, saying it's fewer than 1 in 50,000 people have the disease. Again, what does that really mean? So there's this misconception, I think almost a branding problem of rare disease of like, oh, it's rare, so it's not very common, but it's actually collectively incredibly common. There are over 10,000 rare diseases and counting, and then a vast majority of them are actually genetic. More than 300 million suffer globally, and many in developed countries, but many more in undeveloped countries, like Durhan was mentioning. And then you have this disproportionately affecting children, with 3 out of 10 children not even living to see their 5th birthday. So what do we do about them? Unfortunately, it's not a lot. And fewer than 5% of these diseases have FDA approved treatments. So the way I think of it is like these are ultra rare, but they're collectively common. A litmus test is we probably all know somebody who has a rare disease in our daily lives. And that should be a litmus test that it actually affects so many more than more people than we appreciate. So now the question is what we do about them. And in some ways, the definition of rare and ultra rare, it has a functional consequence of why is it hard to develop drugs for these diseases. So on the right, there's this plot that I'm showing. It's how many people on the y-axis, and then you have what diseases they have, sort of sorted of the most common diseases are on the left, like your Alzheimer's, your Parkinson's, heart disease. And then many of these ultra rare genetic diseases fall into this long tail. Only a few of them actually will have treatments developed because they are commercially viable enough so that a company can commit the significant effort that it takes to develop a drug and time to then make it worth their while from a business sense. So I sit within industry and we commonly go through this problem of, hey, what can our genetic technologies, what can our treatments tackle? And unfortunately, that number of how many patients you need to be eligible for your treatment is around 1,000. That leaves a ton of patients without opportunity, even if the science can enable it. So what we really are faced with is that if we do business as usual, if we do the status quo, we won't have more treatments. It's this long tail that we need to really think about addressing for health equality and just better health outcomes. But doing so actually requires us to think differently and break that mold of drug development instead of just industry taking the charge and economic models. We need this close collaboration between patients, their caregivers, academia, and industry. So why I'm particularly excited about some of the technologies that's emerging is that many of us on this call actually have experience with what are called these platform therapeutics or like platform genetic medicines. And a core concept in how do you approach regulation as well as how do you approach whether you develop whether you give a treatment or not is benefit risk. In this case, the benefit risk ratio using these platform genetic medicines, you might have heard of things like CRISPR or antisense oligos. You actually have some really favorable things. I'll take you through a brief math exercise on here. So if you think about benefit risk ratio as a fraction, right? The numerator is benefit and the denominator is risk. On the top, you have really strong evidence for benefit. Some of these technologies today actually enable us to convert a sequence that is mutated that causes disease into something that is the wild type or something that a sequence that 8 billion people around the world have. And there's this expectation that when you change this root cause in early enough in life, in the right organ systems, you can actually really benefit patients. And then you have the unmet need, right? So many severe diseases just have no treatments right now, otherwise would kill both kids and adults. On the denominator, when you have these genetic medicine platforms, what we've come to learn is that sometimes you can just change a very small targeting sequence and everything else in that drug is kept the same. That actually enables you to keep very well defined safety parameters and also other you know, other features in drug development you actually know very well, and you're just changing the target. That lowers the unknown unknowns when sometimes you change a a target in drug development. And then lastly, this is both a feature, but also some of the challenges is that these patient populations are very small. So that overall, the risk, if you do have something that is developed and it may not be as safe as you'd like it, it's overall bounded. And it's not something that it's put in the water for hundreds of thousands of patients. So all that means is that we're at this precipice in time where we have these platforms with high benefit risk ratios. The regulatory systems are adapting, but many of them we have legacy regulation. And it's really how do we think about this concept of proportionate regulation that can balance the scientific rigor with the speed and cost to reach these rare disease populations. So with that, I'll stop and really turn it over to the real world example that Becca has performed in the past year.
Yeah, thanks, Winston. I appreciate it. It's a good introduction. And I think it's important to sort of, as we talk about what is N-of-1, where are we, where is the state of the field, to take just one step back and think about how fast things are changing. And Winston alluded a little bit to the regulatory legacies that we have. And it's important to remember those regulatory processes are in place in order to protect patients, but they are being forced to be adapted and to be thinking about an entirely new field of medicine that has transformed even in the 15 years that I've been doing this, right? So the world of rare disease medicine is entirely different now than when I began my training in it. And a lot of that is we couldn't even define the N-of-1s when I first started. So if patients didn't have access, I mean, we now have access to exome sequencing, genome sequencing. Obviously, not everyone has equal access, but we can actually look and find what the genetic causes are of all of those N-of-1 cases. This was not the case a decade ago. And so patients might have been identified to have a clinical syndrome, but we wouldn't have been able to point to the exact genetic change that was causing that patient's symptoms. So I will say that widespread accessibility of genomic testing has really transformed our definition of what rare and ultra rare disease is, and enabled this entire field of N-of-1 medicine to even be a possibility. So I think it's really important to reiterate that you have to identify the patients if you're going to try to help the patients. And being able to identify patients is not equally accessible to all. And so that's something that we need to keep in mind. But once you identify a genetic change in a patient, in my clinical practice, I do this all the time, we're really at an exciting time where we could start thinking about, okay, what do we do about it? Now that we know what the exact change is that's leading to a patient's, symptoms or disease or risk down the road, can we do something about it? And we're at a time where this is actually becoming a possibility. And so we talk about platform therapeutics, and really these are a number of different therapeutic modalities that you essentially tune or tweak or repurpose to meet the needs of an individual patient by targeting their own specific genetic change. And it takes effort, obviously, but this is all building on amazing, basic and translational science work that has been going on in the lab, again, over the past 2 decades, that have gotten us to this point where we can harness all these amazing technologies, whether it be ASOs or gene editing therapies or gene replacement strategies, and actually try to move them into the clinic. And it's not ready for prime time for all diseases and all N-of-1 conditions at this point. But I think it's important that we push forward those use cases where there's a possibility of seeing if these types of approaches help a patient so that we can learn, because it's not straightforward how you do this. I'll be honest with you. It's challenging. And we have to go through the sometimes painful process of learning how to actually move these transformational technologies from the lab into a patient. And we're going to do this over and over again to learn how to adapt and do this appropriately and quickly. And then also to give our regulatory colleagues information and data so that they can also transform the regulatory approach for these cases. And so the case Winston was speaking about is an effort, like I said, that we've been developing in collaboration with Kiran Musunuru, my collaborator at the University of Pennsylvania, and many, many others, to develop personalized gene editing therapies for liver diseases, specifically liver metabolic diseases. So these are kids I have the privilege of taking care of clinically, where essentially they have a genetic change that breaks a biochemical pathway in their liver. And so the case that has been recently reported, we knew within 48 hours of life that based on his symptoms, he was very sleepy and not feeding well, and based on biochemical results that we were able to get very quickly, that he had a very severe form of a metabolic disease called a urea cycle disorder, and that we needed to act quickly if we were going to help him. And so we being really my clinical colleagues here at CHOP and Penn, did an amazing job giving him the gold standard care and trying to help him have his urea cycle, which is a cycle important for breaking down protein, function properly, and to protect him as much as we possibly could. But in parallel, Kieran and I and a large group started working and thinking about how do we actually try to correct the underlying cause of his disease. And we could only do this because on by day of life 7 or 8, we knew what his genetic diagnosis was. We had done whole genome sequencing on him, and we could pinpoint the exact spelling change that caused his problem. And so at that point, it really became a race to see if we could adapt, in this case, one of these gene editing technologies, to be able to target his specific spelling change, and correct it. And for the next 7 months, 8 months, it was an all-out sprint to show in a lab that in cells and in mice and eventually non-human primates that this worked and was safe. But also just the process of trying to figure out, how do you manufacture a drug for only one patient, and how do you pay for it? And what's the regulatory process look like? And what are the ethical concerns, which for me is something that I really worried a lot about. And so in our case, I'll be honest with you, it was an all-out sprint, and the stars aligned, and a lot of people donated their time and effort, and things sort of all lined up to be able to, by the time that this patient was not even quite 7 months old, we had a medication in hand and permission to give him his first dose of this medication. And thankfully, it was well tolerated. He received 3 doses of the medication. There were some early signs that there was a benefit, but again, with an N-of-1 patient, it's hard to fully interpret what how big that benefit is. And so I think that's another challenge we face in the world of N-of-1 medicine, is how do you measure efficacy of these new therapeutics? And so this case has obviously gotten a decent amount of attention, and people are trying to figure out how we go beyond this. And I think that's a really important thing I want to discuss today with this group, because it would be a shame, it would be a failure if this is the only kid that ever receives this type of experimental therapeutic, right? We wouldn't be doing justice to this patient, to the system, to anyone. But we also know that we can't do it this way every time. We can't have the stars align and essentially have fate or luck help us get to that point. And I will tell you that while this was a very exciting and, uplifting story to read, I have lots of patients and other patients around the world that this was actually really hard to see. Because if you think about it, why not my child? Why didn't my child have access to this? And some of this is technology. Maybe it's not ready for whatever the indication is or whatever the underlying disease is for a certain patient. But for some of these kids, the technology is ready, and we just don't have the infrastructure to make it happen. And so I think we have an obligation as a community, and I feel a very strong obligation about this, to figure out how we democratize this and how we expand it and how we make it available to others. And I think that's, in all honesty, the real challenge that we face now as a community, how you expand this and pay for this and make sure it's safe. So I'm happy to talk about any or all of those issues as we go along.
Thank you so much, Rebecca, for describing this miracle story. And as you say, it's not just a clinical success, because it is a success even if we don't know further results, but it's an infrastructural success. Because when you were talking about, I was thinking, okay, what if in a hospital in the UK you get a kid like that? Can they bring all the resources together? What about France? What about another hospital in the US? So as you say, a lot of stars align. So Dan, I'd love to hear your comments.
Yeah, I mean, thanks to Becky and Winston. And I think you set the scene brilliantly. And I think, I mean, as you said, it's a miracle, but actually what we need to do is we need to scale that miracle, and we need to be able to ensure that all the children that are diagnosed within their therapeutic window, that there is an opportunity for them to also have treatment. So I think, Winston touched on it at the beginning around, proportionate regulation, but that's not the only part of the ecosystem we need to change. And I think, hopefully we'll come on to discuss it, because that end-to-end pathway is really, really crucial. And that visibility in the end-to-end pathway and building that infrastructure for patients to link the diagnostic part to the actionable mutation, where there's a modality that will work and hopefully correct that mutation, and then sort of build that into the health system, both from an access perspective from regulators and sort of payers, and then follow up. So some of the work we've been doing in the UK, which also involves multi-stakeholders, including industry, which can sort of bring expertise and sort of scalability, is trying to look at that infrastructure in a way that is sustainable and scalable. And that's through the Rare Therapies Launchpad. And we're working initially on a single modality. So we're working on oligonucleotides, but the idea is to create a pathway that will be open to, which would certainly be sort of condition and modality agnostic. And I think, I mean, when you listen to the work that's gone into it, when you say it's not easy, I mean, that's an understatement, isn't it? When you think about the problem statement. And so that's why, but what I am really heartened by is that there are, there's the RTLP in the UK, but also other initiatives, Winston's work in the States, Becky's work in all across the globe. And I think the interest in trying to solve this as a societal, academic, scientific, clinical problem, I think is really aligned. And I think there's huge opportunity to ensure that we don't kind of miss out on 21st century science, but it's going to take a collective effort, and it's not a single person or single infrastructure that's going to do it. It is everything from data collection. And one of the things we're really trying to solve in the UK is, when do you have that switch from research to treatment with iterative learning? So there's a point of the maturity of that modality, that technology, where you say, well, actually, in areas of really high unmet medical need, we need to do better for our patients. We need to move on from saying this is research, and we need to then flick that switch and say, actually, this is treatment first, and then it's learning second. Learning's always going to be really, really crucial, and learning from each patient is really, really, really crucial. But there is that, collective work from different stakeholders within the ecosystem, I think, is going to help solve this.
Thank you. Durhan.
Yeah, I really want Carol to answer some of this, but I thought I'd jump in. Maybe she can answer both of us. But I just want to give a different example and some of the barriers that we're running across in terms of the regulatory. And, an example of a parent actually in Canada who had a child with, a ultra rare condition. In fact, it was the only one he knew at the time, raised, somewhere around $4 million to get Toronto sick kids to help develop an AAV gene therapy. Took 2 or 3 years, and they've administered the therapy to this child. But at the time they did it, they asked, can we produce multiple copies of this therapy? We've got, as you say, the platform, we can do this, with the goal of giving the other therapies. Way at the time, over that 2 or 3 years, they'd identify a handful of other families, 5 or 6 families, and that's why he went to 6 to begin with. And he promised them, he said, come and join us, and share your information, and we will give you the therapy. As it turned out, it was not so easy to give this therapy away. He's in Canada. I think the other patients were all in the US. They had to figure out how they were going to actually get through some kind of that regulatory, licensing or review of what it took to kind of get the therapy beyond the single case. Setting up a clinical trial was kind of the answer, but even that was not straightforward. And right now, of course, they're providing, I think, at least a couple of the children through the clinical trials. The sad thing, I think, is that it's taking so long to do it. Some of the children who might have been eligible at the time they all started, this may be no longer quite likely to benefit as much as they want, or even benefit at all. So I hear what, both Becca and Dan are saying, how do we kind of scale this up? How do we do it faster? But I think what we're learning painfully one by one is that we're not really, we don't have any mechanisms that allow us to do that, right? So it would be lovely if the regulatory scientists were actually saying, okay, this is where we need to go, this is where we want to go, can we create a different kind of a regulatory framework? Because we are finding, even with some of the CAR Ts that we're doing in hospital, bam, trying to get it beyond the hospital, it runs into, if you want to make it available for more than your kids and you want to so-called commercialize it, here's all the things you need to do, and we can't do it. So I think, recognizing this is a whole new area that we're talking about. Can we rethink a regulator? And don't even get me started on what HTA would do. I mean, I just came back from HTAI in Buenos Aires, was at ISPOR 2 weeks ago, and we haven't got a hope of how we're going to approach this in any way of making it commercially available, marketable, etc. So I would love to, and what I plead for at HTAI is, can we start over? If, in fact, we think about the therapies that we're trying to get through now, we would never have invented what we've got now in either the regulatory or in the HTA space. So can we think anew? And I think that's kind of my plea, in terms of trying to get it faster, better. Can we, we'll not start from absolute scratch, but kind of get beyond kind of what we know as the current process, and not just trying to modify the current process to fit what is truly novel problems.
Thank you, Carol. No pressure, but it's up to you to offer the.
Where do I start? That's one of the issues. So as I've been listening to this and thinking about this for a bit, it does sort of blow your mind, doesn't it? Really, because every single bit of the system, if you take a look at it bit by bit, none of it fits for this type of problem. Absolutely none of it fits. And certainly, Duran, the HTA side of things doesn't fit for lots and lots of reasons. You know, normally HTAs is taking a lens on a sort of very wide perspective, looking at the healthcare system, looking at costs and benefits. But when you have a technology that is literally just nothing like you've seen before, which is this, and the term I'm going to use because I think it's really useful, labels sometimes do work, and I think it was you, Winston, interventional genetics. We have a completely new modality of treatment, nothing like it before, and as a consequence of that, the health technology assessment world probably doesn't even yet got a grip on what the intervention really is. And if HTA doesn't have a grip on the intervention, it can't really do an evaluation. It'll have a grip on the population. That's really straightforward. It's an individual. It'll have, so I'm using the PICO process. It has a grip on the comparator because that's nothing else. Wouldn't be doing anything else of this nature. And it's pretty much, I think, got a grip on the outcome, which is, you know, with these types of conditions, the survival rate is extremely, extremely low. So you're probably looking at some form of survival outcome. What you don't really know is what the intervention is, because it's not the individual. I don't think it's the individual change in the gene. It's the platform. And maybe that's going to be the way in which we can start to wrap a little bit of objectiveness around it. Because if you take away the individual and think about the platform, you start to realize the types of methodologies and the types of change in the way that you look at the intervention from an HTA perspective. It's probably going to be the same in the regulatory environment. They're probably moved a little bit further than the HTA people have. But if you can start to evaluate perhaps the characteristics of a platform system that need to be evaluated, if you can start thinking about how you can benchmark what good looks like on a platform technology, all of those sorts of things that HTA can get a grip with, you could probably start to develop a whole new way of thinking about these types of interventions, these genetic interventions. So that's where my mind's going. The other issue with HTA in many places is it is trying to balance, as I said, the benefit and costs. We haven't really got a handle on what sort of change this is going to do to the cost base of the healthcare system. You know, it is going to completely change probably the priorities of that. If you're thinking about N-of-1 not being N-of-1, but being that long tail, you've got a massive system that at the moment there's nothing for, that there's now the potential intervention for, and you've got a healthcare system that can't really manage at all. So you're now starting to think about how you need to configure the entire healthcare system, and therefore probably develop again, I'm coming back to HTA methodology, develop an HTA methodology that is much more embracing even than the current health economic approach to what that system change needs to look like, and also the evidence you need. So I am probably talking about the most hugely complex problem that we face at the moment. I'm thinking that, like Dan, the collective effort is the only way we can do it. We can't have the regulators and the HTA people going off and doing their own thing. They've got to come together, and they've got to come together in a way that is resonating with the rest of both the healthcare and the patient community. So it seems to me that this isn't a problem that any one stakeholder can solve.
Thank you, Carol. Winston, please go ahead, and then I have a question for Rebecca.
Yeah, so I actually was going to just say the UK has really taken some of the lead on this, and that's thanks to the work in the RTLP that Dan and colleagues have done, where there is, so I guess if we step back a bit, we see this inherent challenge of regulatory authorities. I've heard this over and over again. It's like they learn alongside the sponsors with every new program that comes in, right? And I think that's fair, right? They're in collaboration. There's a lot of engagement. The question is then, how do you close the loop to update regulatory policy? And that seems to be actually a slow loop where even if regulators learn things, you hear anecdotally that now it depends on the regulator that you get. It's not consistent. Sponsors offer up different things and regulators just say yes, because like, why not get more data if a sponsor is offering something more than you need? And so I think this is where the Rare Therapies Launchpad, there's a very thoughtful implementation of saying there's more of like a protected sandbox. And Dan, I'll let you describe more of this, that the thinking, or really that some of the learnings can happen with almost like a decentralized with appropriate oversight, like more self-regulation based on the folks who are the closest to the science and closest to the specific implementation details that are important. And then maybe my second comment will just be around, we're not innovating in a vacuum, I think, and we're also not innovating based on things that have not been done before. The wording of interventional genetics is actually intentional, where it's like our surgical colleagues, right? People who
Absolutely.
are on the other side of medicine versus surgery, they've been doing a lot of that balance of research versus treatment with iterative learning. Colleagues in oncology as well. Like there's a lot of models that we can say of how can you learn by doing, and doing so in still a way that protects patient autonomy, protects the really fundamentally the patient. And I think borrowing some of those concepts and seeing how we can now apply it to complex drug development on these platforms, that's something we should try to not reinvent the wheel and instead both adopt oversight models as well as reimbursement models eventually too.
Dan, please go ahead with your comments.
Yeah, just before you're going to ask your follow-up question, just to build on what Winston said about the RTLP, I think there is an opportunity, and it's not the only opportunity, but I think what we want to look at in the UK is really to test that model through a pilot to kind of think about what are the guardrails for treating patients in centers of excellence that are used to kind of working in challenging clinical scenarios, but to provide them with that additional supportive sort of voice, if you like, that you don't currently get at the moment, where if you look at sort of what Tim Yu has done, for example, he was drug developer, he was a whole suite of all of the different sort of roles and responsibilities that you would allocate out to the whole suite of people. But actually what we want to do is to provide them much more supportive environment for that switch between treatment and research and treatment with iterative learning. And part of that is to kind of consider a framework around accreditation, where the collective health system says to a center of excellence, actually, we have confidence in this modality and we have confidence in you delivering this modality for a suite of or set of patients within particular types of conditions. And then you do and tell type regulation, where you tell the ecosystem, but you ensure that you have, central data collection so you can feed that back into the ecosystem. The problem with doing this on a kind of case-by-case basis in terms of compassionate use, which is an extremely useful tool, don't get me wrong, I'm really supportive of sort of compassionate use to named patient approaches, but unfortunately that data often gets lost and isn't shared into the wider system. And then that's a problem because we don't learn as much as we should do to support the next patients kind of coming through. So I think that accreditation center type model is something we'd really like to test. It's not going to be for everything. It's not kind of like trying to break up the entire rare disease ecosystem and say everything goes through accreditation, because we know actually there are many common rare diseases where actually a traditional approach to drug development works perfectly well. But there are those areas, as we're talking about today, where we absolutely need a new model, and we really have to think about utilizing some of the principles and procedures that we already have in place and reworking them. Where do they add value? And then ensuring that we bring them in together in a collective in terms of a new end-to-end pathway.
Thank you, Dan. Carol, and then Rebecca.
Yeah, I'm building on some of Dan's comments, because one of the things that over the years I've been perhaps a little bit frustrated with, but it's not going to happen with this particular collaborative, is that we have lots and lots of conversation about the regulatory world, and then, oh, we need to think about HTA somewhere over here. And that creates a massive divide between the pathway from innovation to access. And one of the things I've been, as I listen to Dan, I'm sort of thinking that the concept of HTA in the context of that type of process that Dan has articulated is much more about understanding healthcare system impact of the switch between the, in a sense, the experimental to the clinical. So just follow me, because at that switch, there is a use of healthcare resources. And as a consequence of that, there's an opportunity cost for the healthcare system. And so we do have to spend a bit of time thinking. We've got a fantastic system accreditation. We've got systems and processes that make the modality feel, have a good benefit risk ratio. But if we could then plug in, how do we ensure that we've got a healthcare system that can function with that type of modality in a sustainable way? We might actually start to develop a completely new concept of the way in which HTA can be used in the real world in a proportionate way. When we've talked about HTA previously, you know, proportionate HTA, it's about shaving off the bits and pieces of the standard HTA process to make it a bit quicker. But proportionate HTA in this context, I think, is a completely different way of thinking about what particular unique aspect can a health technology assessment lens bring to this. And I think it's about the use of healthcare system resources on the ground. Just a thought, throw it out.
Thank you, Carol. I think my comment partially connects to yours and gives a chance for Rebecca to respond. So my question to you, Rebecca, is how scalable is the treatment that you have developed? And not just for this particular treatment or the types of treatments, for example, Melastin, or the ones who were designed for that patient. Of course, this is not the pill that we can ship elsewhere, but how transportable is that learning? And the reason I'm asking that question is, if we figure out the way of scalability, sharing with other centers, kind of creating, at least technically, implementation to more than one patient, then HTA question can focus on scalability issue. Even if it's very few patients, the methods will be the same. It's just, but then looking at the problem that you've solved or attempted to solve, this is drug development. And the thing is, who funds drug development and who funds the scaled products after we found a solution, even if further modifications are needed? So these are two very different economies. Both of them seem to be very expensive because industry at the moment is not part of the equation because you did it in the hospital. It might be part of the equation depending on the condition, right? So the issue of patents arises, and then it might create even further financial complication here. So if we split the problem into two, so who funds what, and can we even look at it this way, or maybe we can't?
It's a great question. And I'll be honest, there's not a clear answer to this. In terms of scalability, in terms of going to different sites, I think parts of the process are very scalable, right? I think it sounds crazy, but some of the basic science or the initial finding and editing solution in our case is the easiest thing to transfer and scale, right? That science is pretty well established and could be done lots of places. The manufacturing is a bottleneck, and you identified a huge part of that bottleneck, which we experienced, and I've been very open with it, is the contracting and the negotiation and the sharing of IP and all of it's silly because, it's an N-of-1 case. This isn't going to be used again. But still, contracting was a huge hurdle for what we tried to do, because while we actually manufactured with amazing manufacturing partners at Aldevron, the Danaher Corporation, so Aldevron, IDT, Acuitas. But some of those contracting negotiations were the things that were going to sink us the most. I'll be honest, it was the hardest obstacles to overcome. And so manufacturing right now is not scalable, but I think we can translate into a system where it could be scalable. And then administration of these therapeutics could be done at a number of sites, but you would want teams in place that understand the disease and understand the needs of these patients, because there are going to be unintended side effects down the road. There are going to be adverse events. We're learning this in the rare disease space all the time, and you need to have expertise at your clinical center to be able to respond to those. In terms of scalability and who pays for what when, I like Winston's discussion of an interventional geneticist. I will tell you, it is the hot catchphrase in my field of medicine. Every young physician scientist or physician that interviews at our program wants to be an interventional geneticist. It is a hot topic. And if you really take that analogy a step further, if you have a surgeon innovating in a new space, you don't ask, and maybe they use a different form of a scalpel, this is very simplistic, but a different tool to complete that surgery than what is traditionally done for that surgery. You don't ask who's paying for the scalpel, right? Like you realize that is part of the healthcare system. And so when we get to a point, if we're just changing the tool to do the genetic surgery, and in this case for gene editing, that's all you're doing is you're changing essentially a guide RNA, which is the little GPS signal telling where the edit should happen. If you can do that safely, perhaps through an accreditation process to show you so that you know what you're doing with choosing which scalpel, choosing which tool to use, really, that shouldn't be any different than reimbursing for surgery, because that's essentially what you're doing. You're attempting to make a definitive correction or definitive change using a tool or using a technology. It's just, in this case, it's a very somewhat complicated technology. And I will say one last thing, sort of to some of the points that were raised earlier about other models where this has been done, and moving towards an accreditation type model. If you look in the field of laboratory diagnostics and molecular diagnostics, this is done all the time, right? There are many, many, many laboratory created tests, laboratory developed tests, and especially in the genetic space, that are developed at academic centers, that are developed at labs, that are used as part of clinical practice. It sort of moves that across that gulf from research to diagnosis to clinical diagnosis, reimbursable clinical diagnosis, without ever getting any regulatory approval. But the labs have to go through, in the US, a CLIA CAP certification or an appropriate certification to show that they know what they're doing. And so I think it's a weird hybrid, this field of a surgical intervention, and in all honesty, some of these new innovative technologies that we can learn from our clinical pathology colleagues, and some sort of hybrid version of those two things that might get us to a point where we can wrap our head around the structure. But I'll stop.
Thank you, Rebecca. I'll give a word to Winston in the end, and then I have a question for Duran.
So I love thinking about this because I think, one, you're absolutely right that the industry participation is necessary. And just to echo Becca's point, when a surgeon is using a tool that is like, say, a new catheter or a new stent that's being placed, maybe once upon a time that was soldered and engineered in their laboratory, but by and large now these are all provided. Even like 3D printed heart valves and stuff, they're provided by a manufacturer who has the scale, the ability to do this. And I think that's where some of the, you let the market forces help drive the innovation and the cost down. Because to Carol's point too, we have to think about what are the impacts on the healthcare system. If every treatment is going to be $2 million, that's actually not sustainable given the number of rare diseases out there. But we're in this weird chicken or the egg problem where, because there aren't scalable, fit for size or fit for purpose approaches, the the treatment for baby KJ, didn't you guys make like 10x the amount that was necessary because that's not scaled down? That's expensive. That's wasteful, yeah. Right? And I think this is where when we get this process that's going on repeat. So if I zoom out a second, I'll just give you some real numbers, right? a lifetime supply of antisense oligo for a single patient, like, drug grade, can be anywhere from $20,000 to $50,000, right? It's like, a mid-sized car. The challenging, there's other parts of it that are not fit for scale, which is like now aliquoting it into specific vials, like literally into small vials, the fill finish, that can be $200,000. And then toxicology packages can be hundreds of thousands of dollars. So that's not the case for CRISPR. Actually, CRISPR, in some ways, a lot of it costs a lot to actually make the drug itself. So you're in this place where the economies of scale for oligonucleotides have been driven down by the success of drugs like Spinraza, now ASOs, as well as just for, people doing decades of DNA synthesis work through companies like IDT. CRISPR has been these lipid nanoparticles that help get to the liver, as in the case for baby KJ's treatment. That has also been driven down by mRNA vaccines that use lipid nanoparticle technology. Like Acuitas was actually one of the makers of the Pfizer vaccine, and also mRNA. So those are all helping, but the more that we can get this flywheel going and actually have the right fit for purpose resources, I think you can start compressing that cost so that it's not just this crazy expense that's going to be incurred by the healthcare system, and no insurer and say private insurers in the US want to pay for it. Yeah, so there's lots of thoughts here to continue, but just it's a really important question to think through of how do we get that flywheel going.
Thank you, Winston. Dan.
Yeah, just quickly, as I see we're running out of time and you've got some additional sort of questions for others. But no, just building on the reimbursement part, and I think there's two elements to the funding aspect, and certainly that's something we want to look at the RTLP. And I think the first one is not to forget that there'll be activity costs for the centre. And particularly if you're sort of switching from research to treatment, there'll be some elements of research, such as research nurses and things that are traditionally part of that kind of process to support the delivery of that modality. So I think it's really important we think about, modeling some of that reimbursement and funding is A, that there'll be reimbursement or funding needs for the centres of excellence, the accredited centres, but B, there's also going to need to be some form of funding for the manufacturers, who hopefully allows the scalability of that. And building on what makes this scalable, I think what makes this scalable is having a visible pathway that is collectively agreed by the health system. I think that provides a really important demand signaling from the system for companies to say, look, actually, here is a visible pathway for you to follow, because at the moment it's so difficult. So that's why it's really hard for companies to kind of engage with that. But I think if we do create this visible new end-to-end pathway, it's a lot easier for companies to understand where they can actually move into that pathway and then support the delivery of medicines at scale using their experience, but also being able to deliver that in a way that is hopefully at scale because of the capacity that they have. So I think those are the two aspects that we have to consider from a funding and scalability perspective.
Thank you, Dan. Durhan, I have a question for you. Probably not an easy one, but putting a global hat on, if you had no resource limitations and no political resistance, it's not exactly for this world question, but for example, if you had complete freedom to design either a system or an organization that would take charge of this problem, would you do something from scratch? Would you give the responsibility to an existing organization or the network of existing organizations? Because one very clear theme here is if we want to achieve success, we must collaborate. We must create these platforms of sharing knowledge, experiences, samples, data, everything. So, and hopefully maybe economies of scale in terms of costs and price, that would be very, very ideal. So what solutions would you come up with?
Well, I don't have any solutions, but listening to what Dan and Carol and Winston and Becca are talking about, I get this vision, let me just say, of a big bubble that would be research and actually for simplicity, right? Scaling to implementation. The challenge at this point is that bubble is not one bubble. There's a research component which is funded through certain means. And even when we're working with, for instance, a number of small companies that are developing, which are not necessarily N-of-1s, but N of a very few, let's put it that way. And again, at the research end, there's investment into it. And the investment comes because they're hoping they're going to be able to generate something at the tail end when it becomes marketable. But the problem is, in fact, even in the kind of models that Dan or Rebecca or Winston are talking about, it requires then the health system, as Carol says, to actually invest in it. And quite frankly, when you start to look beyond kind of just that particular technology, you're running against the rest of the health system. I mean, if we had nothing to do but small patient populations, and we had a dedicated budget for small patient populations, not a problem. But my health system is constantly looking at what else could I do with that money, and it's not just in that small bubble. So I guess one of my questions would be, if we're thinking about beyond US, beyond high-income countries, would you create a bubble that would also allow us to think about, providing access to a broader population that their health system is not going to be paying for, that there is going to be investment all the way through that that whole chain. So yes, investment in the research. I mean, it's kind of Winston's notion. The difference between something like a scalpel and a drug is that we don't expect the scalpel to really have to go through a separate HTA. Nobody's going to go to your health system and say, geez, what's the cost effectiveness of that scalpel? But if you suddenly transform it into a gene therapy, suddenly it becomes, right? And as Becca's talking about, the individual components of that gene therapy, nobody's going to ask. But once you make it into a therapy, suddenly somebody's asking. So I'm wondering if in this very experimental stage that you've created, can we not create a bigger bubble where all of it's experimental, including the access to the patients and that where your funding comes is that it has to cover the whole thing. I mean, that's a little bit idealistic, but it would also allow us to say, okay, where have we got communities? I mean, I'm looking at Indonesia, which is really ramping up their genomics. I'm looking at places like, China and, Taiwan that are investing in this big time. Can we create a bubble that would include them as part of that research component? And it may be just totally unrealistic, but to me, unless we can cover the part that's going to provide it to that larger group of patients, it's never going to happen.
Thank you, Duran. At least we have the name for it now. It's a bubble. So fantastic discussion. I very much I learned a lot, and I very much appreciate your expertise. And we've been so busy listening to your experience that we didn't even have time to ask our audience to come up with questions or contribute. So that tells me that we found a very prolific topic, and we just did an introduction to it. So there's a book to be written. So thank you so much for your time, for sharing such insightful information. Lots to think about, and for us at Consilium, perhaps to think about how we can take this discussion further and stay on top of the developments. So best of luck to Rebecca and Winston for developing these technologies and changing the world. And to Dan and Carol and Durhan making it happen. So letting patients actually see that. So thank you so much, and we'll see everyone on the 10th of July. So it's a little bit of a break, and we have a very interesting talk for you about the FDA drug approval. All right, have a good afternoon, and
Thanks
thanks for
very
joining.
much. Thank you.
Bye-bye.
Bye.